Hi viewers, today I am going to discuss a new topic, that is new molecular methods for the detection of waterborne organism, or the recent techniques used to detect the pathogens present in the water. Okay. We have already discussed many methods like MPN or the most probable number method, defined substrate method, biochemical test, etc. These cultural methods are not accurate. So the novel techniques are introduced to study the aquatic microflora. The recent techniques include flow cytometry, epifluorescent microscopy, then transmission electron microscopy, nucleic acid technology, nucleic acid hybridization, and finally PCR, or the polymerized chain reaction. Flow cytometry. It is also called fluorescence activated cell sorters. These are instruments which analyze optical parameters of many individual cells in a short time. Okay. So it measures the optical property of the cells in a sample. The sample flow through an orifice and across the path of light source. Usually a laser, optical parameters like light scatter and fluorescent are measured on each cell by photoreceptor. So, the light scattering and also the fluorescent property of each cell is measured by photoreceptor. The signal received are processed by computer on the basis of optical density of the cells can be sorted and collected automatically. There are several advantages for this method. First one, rapid analysis of large sample size, that is 10 raised to 4 to 10 raised to 6 cells per minute. Okay, that is the capacity. Second advantage, it can detect small and less abundant cells in natural water sample. Another important one, analysis of natural phytoplankton and autofluorescence. Okay. Then cyanobacterial phototropes are enumerated by this method. We can separate cyanobacterial phototropes from phytoplankton by this method. This separation is based on phycoerythrin and chlorophyll fluorescence. That is, cyanobacterial phototropes fluorescence orange color and phytoplankton produces red color. So we can separate cyanobacterial phototropes and phytoplankton by flow cytometry. Okay, or the fluorescence activated cell sorters. Epifluorescent microscopy. This microscopic technique used to count direct total count of the bacteria. It employs fluorescent stain or fluorochrome stain. Example for the fluorochrome stain is the acridine orange. One of the important new example for the fluorochrome is the 4,6-diamino 2-phenyl indole. Okay, that is indicated by DAPI. Once more, 4,6-diamino 2-phenyl indole. When using fluorescence technique, the sample is either stained and filtered onto membrane filter or cells are stained on filters. Fluorochrome stained cells help to count bacteria in the water. Most of the microorganisms present in the water is in active state. So to get an estimate of metabolically active cells, a dehydrogenase enzyme stain assay is used. In this, active dehydrogenase reduces a synthetic water-soluble membrane-permeable straw-colored salt called tetrazonium salt by converting to a pink red. Okay. And the pink red compound is called formazan. This formazan represents metabolically active cells. So the dehydrogenase enzyme reduces the tetrazonium salt to formazan and produces a pink red color. And the formazan is metabolically active cells. Okay. Another important method used for the detection of waterborne organism is the transmission electron microscopy. It is a type of electron microscopy and it is used to count the bacterial number. Okay. Transmission electron microscopy used for the biomass estimation. One of the disadvantages of transmission electron microscopy is the difficulty in producing quantitative preparation. And the transmission electron microscopy provide more information on bacterial morphology than the epifluorescence technique. And it provide direct counting of the viruses present in the water sample. Okay. It is the only method for the enumeration of the viruses. Its efficiency is dependent upon the concentration of virus. Okay. It is determined by flow filtration. Next method is the nucleic acid technology. Here, characterization of natural population without isolating or cultivating the individual members possible. But it requires purification and concentration of nucleic acid from microorganisms. Then the next method is the nucleic acid hybridization. Molecular probing is used to detect genes of specific bacterial metabolic activities such as heavy metal detoxification, aromatic or xenobiotic degradation, and pathogenic bacteria. Nucleic acid hybridization involves denaturation and immobilization of DNA or RNA from water microorganism onto nitrocellulose membrane or other membranes. Okay. That is, the DNA or the RNA required denaturation and immobilization. Okay. PCR, that is polymerized chain reaction. It is an enzymatic multiplication of specific genes in vitro which involves a thermostable DNA polymerase. This can detect the organisms or pathogens against ambient microorganisms. And also it can detect conserved genes in the indigenous microbial community. PCR technique used for the detection of Legionella pneumophila from water and also for the detection of coliforms present in the water. For the detection of coliform, LAC-Z gene is selected as target site. In addition to the LAC-Z, MAL-B and LAM-B are also used. MALB code for maltose protein and LAMB code for surface protein of E. coli lambda phage. LAGZ detect all coliform, but LAMB or the surface protein of the E. coli lambda phage detect E. coli coliform only. Okay, so that is the difference between LAGZ and LAMB. So by this method, both culturable and non-culturable coliforms can be detected. Okay, so that's about new molecular methods used for the detection of waterborne organism. Okay, thank you.